moving on. All right, let's see where these proteins go. Now we're on vesicles. So remember, we're kind of getting into more cell biology. So now we're going to look at what these proteins are doing. But I don't want you to forget all the regulation, right? How we got here. I know there's a lot, but you want to keep in mind that cells don't express all proteins all the time, right? So those differences then are going to lead to the differences in the cells, their characteristics, how they function, and so on and so forth. Okay. So do you think it's important that proteins get to where they're supposed to go?
Of course, 100%. Where they're useless.
So tell me, where are the glycolytic genes? Where do they end up? The proteins end up?
Outside the mitochondria.
Which is called the
Cytoplasm?
cytoplasm. No, outside the mitochondria. Come on, glycolysis is in the mitochondria. No, it's not. It's in the cytoplasm.
Right outside the mitochondria.
So if you think about it, when those genes are expressed, and they're pretty much constitutive, so they're on all the time, those ribosomes are going to be making those glycolytic enzymes right out in the cytoplasm. They're not going to be on the ER typically. Because then what are those proteins? They fold, right, after they're done with translation, and then they just start doing their jobs right away. Now, other proteins, especially proteins that are secreted, so outside the cell, they're going to have to go through the ER. So pretty much 100% of them. Okay, so anything that has to be modified, because either in the ER and also in the Golgi, we're going to see there's lots of different things that can happen to a protein even after translation. So we're going to see here, even post-translational modifications can increase the diversity of the proteins we can get. I think we've already seen some of that when we looked at protein expression or protein function. So I think it's an obvious question that these proteins have to get to wherever they're going. And so what we're going to see, there's a whole endosome systems, what they call it, where through vesicles, these proteins can be transported to different areas. And as it turns out, the proteins themselves kind of have an address label. Something on the next one. Yeah. A lot of them have an address label or a signal sequence, which tells the ribosomes and other translation factors where these proteins are supposed to go. So obviously you don't have to memorize these, but it's kind of cool to think about that you can actually put a signal sequence on these things so they get to where they're supposed to go. Many times the signal sequence is also cleaved off after they get there. So it's kind of a temporary little label to put on there. It's kind of cool. So what are the different mechanisms to get to where we're going? Well, one, obviously, you're going to use the nuclear pores to get back into the nucleus. So if you're a polymerase or something like that, you're made out here on the ER, in the ER cytoplasm, you can be shuttled back to the nucleus through the pores. The pores are going to have they have proteins associated with them, which are going to be responsible for regulating that. Transport across membranes. So if you're getting into chloroplast or mitochondria, kind of interesting, we're going to see that proteins are actually unfolded typically to be transported across a membrane and then refolded with the help of chaperones. So that's kind of an interesting one. And then through vesicles as well, a lot of those can either be held in vesicles or they can fuse with membranes. So we'll take a look at that if we get to that today. It's kind of interesting. So several different techniques to do it. One, using the nuclear pores. So the pores actually have a lot of different proteins in there, so pretty heavily regulated, where they're going to recognize typically what are called nuclear transport receptors. So there are specific proteins that will help guide you, if you're a protein, across the nucleus, the nuclear pore membrane or the nuclear membrane to get into the nucleus. And then these guys are recycled back out. So is it important to do this?
Yes. Yes.
Is there any importance in regulating this for the nucleus? Got a dumb question, 3/4 of the way through the semester. But have we seen anything that's not been regulated? Is that a dangerous question?
I guess what I'm talking about.
Talk about what?
Cancer, I guess.
That's true. Yeah, we haven't talked about cancers. They're pretty much unregulated.
They'll pretty much see them really regularly. That's not, it's like kind of protein on the-
Well, it's been half.
I mean, for example.
That's true. He's saying delta not.
Slow it down.
That's
Once-
true.
Once it gets inside the cell, there's not.
Yeah, you don't want self-affecting DNA. I like that. So yes, it's quite important, I think, to make sure you're regulating this just like anything else. There's probably a reason. Yeah, why do we have membranes anyways? Why do we have a nucleus? Why do we have a chloroplast? Well, we don't know. Why do plants have it? Why do we have mitochondria? What is the whole point of that? Yeah. You hear that? So compartmentalization in eukaryotes, I guess, is pretty important. It helps us with these processes, making them more efficient. So I think the same thing with the nuclear pore. It's really important that things just don't leak across if they're not supposed to. So you can think about mRNAs that are not properly modified. They're not allowed out of the nucleus. They're destroyed. Other proteins, you just don't want random proteins coming through. So that's why I think it's so important to have these receptor proteins that bind to whatever the cargo is and bring them in a regulated manner. So it's pretty important. Although during the cell cycle, it doesn't really matter, does it, right? You guys know what happens to the nuclear member?
Goes away.
You just destroy it anyways, right? So it seems a little pointless at some point. Okay, so that one's pretty easy, getting things in and out of the nucleus. What about getting across a membrane? This is kind of cool. So here we have protein with its little address label. Those are going to be identified by, what is it, membrane-specific receptors. So on whatever organelle you're dealing with, you're going to have a signal receptor, which is going to recognize that signal, and then that is going to help the translocation process across this membrane. So it's pretty cool. So these receptors are going to bind to a translocation protein. And then typically, I think this is pretty true for every protein, they're going to be unfolded as they come across, and then they refold when they get into whatever the organelle is with the help of other proteins. And then the signal sequence is typically cleaved off. So kind of an interesting process. Do we have to be careful with this? Unfolding and refolding proteins?
Yes.
Is there a rumbling?
Sounds like ceiling.
Maybe it's just the air handling system sometimes. Okay. Don't be alarmed. It's okay. Why do we have to worry about this? Sorry? Yeah, misfolding, right? Misfolding. You want to make sure if you're going to unfold a protein, you have to make sure it's going to be refolded to avoid any issues with that, right? Okay, so here, these receptor proteins are going to interact with this translocation, and then you can kind of see it's a huge complex here. So we have the ribosome that's connected to this, and so they're all work together. The ribosome, the receptor, and the translocator. As the protein is made, it's going to be translocated across this membrane. Now, if it's a soluble protein, what it does is it just gets translocated, and then typically they cleave off the signal. So the signal here is really the only thing that is trapped in the membrane. And then you have your soluble protein. So if this is in the ER or something like that, then it can be worked on or modified, whatever it's going to happen to. But what about membrane proteins? You guys ever seen how membrane proteins are put in here? Yeah. Has anybody ever done any sewing with a sewing machine? It kind of reminds me of that. This is kind of cool. So here, what we're going to see, and we will watch a video on this because it's kind of funny. Okay. So again, you still have a signal receptor, but think about like a, I don't know, a GPCR. What is that? A 7 transmembrane or 12? I forget, usually 7. So you have to have some way to be able to thread basically, or put that protein into the membrane, because it's going to be a membrane protein. And what it turns out, there are actually internal signals that tell the translocation protein where to stop it or where to start it. Typically, what do those intermembrane areas look like in terms of chemistry on a protein? Is remember what is the in the middle of the membrane look like? Is it hydrophobic or hydrophilic? Right. Yeah, so a lot of those signals are actually hydrophobic or hydrophobic. So that kind of tells you. So let's watch a video on this, kind of fun. So I just looked up one from one of the books. If we can have sound. Oh, well, you know, I have to unmute it.
Or ER, is the most extensive membrane system in eukaryotic cells. Proteins transported to the Golgi apparatus, endosomes, lysosomes, and the cell surface all must first enter the ER in the senescence. As an mRNA molecule is translated into a protein, many ribosomes bind to it, forming a polyribosome. There are 2 separate populations of polyribosomes in the cytosol that share the same pool of ribosomal subunits. Free ribosomes are unattached to any membrane. Membrane-bound ribosomes become riveted to the ER membrane and translate proteins that are translocated into the ER. These membrane-bound ribosomes coat the surface of the ER, creating regions called rough endoplasmic reticulum. 2 kinds of proteins are moved from the cytosol to the ER. Water-soluble proteins completely cross the ER membrane and are released into the lumen, while transmembrane proteins only partially cross the ER and become embedded in the membrane. All these proteins are directed to the ER by a signal sequence of small hydrophobic amino acids. The signal sequence is guided to the ER membrane with a signal recognition particle, or SRP, which binds the ER signal sequence in the new protein as it emerges from the ribosome. Protein synthesis then slows down until the SRP ribosome complex binds to an SRP receptor in the ER membrane. The SRP is then released, passing the ribosome to a protein translocation channel in the ER membrane. Thus, the SRP and SRP receptor function as molecular matchmakers, connecting ribosomes that are synthesizing proteins containing ER signal sequences to available ER translocation channels. In addition to directing proteins to the ER, the signal sequence functions to open the translocation channel. The signal peptide remains bound to the channel, while the rest of the protein chain is threaded through the membrane as a large loop. Once the protein has passed through the membrane, it is released into the ER lumen, after the signal sequence has been cleaved off by a signal peptidase located on the luminal side of the ER membrane. The signal peptide is then released from the translocation channel into the membrane and rapidly degraded. It is thought that a protein serving as a plug then binds from the ER lumen to close the inactive channel. But not all proteins that enter the ER are released into the ER lumen. Some remain embedded in the ER membrane as transmembrane proteins. For clarity's sake, the membrane-bound ribosome will be omitted to illustrate the translocation of transmembrane proteins into the ER membrane. In the simplest case, that of a transmembrane protein with a single membrane-spanning segment, the N-terminal signal sequence initiates translocation, just as for a soluble protein. But the transfer process is halted by an additional sequence of hydrophobic amino acids, a stop transfer sequence, further in the polypeptide chain. The stop transfer sequence is released laterally from the translocation channel and drifts into the plane of the lipid bilayer, where it forms a membrane-spanning segment that anchors the protein in the membrane. As a result, the translocated protein ends up as a transmembrane protein, inserted in the membrane with a defined orientation. In some transmembrane proteins, an internal signal sequence is used to start the protein transfer, which continues until a stop transfer sequence is reached. The 2 hydrophobic sequences are then released into the bilayer, where they remain anchored. In complex multipass proteins, in which many hydrophobic regions span the bilayer, Additional pairs of stop and start sequences come into play. One sequence reinitiates translocation further down the polypeptide chain, and the other stops translocation and causes polypeptide release, and so on, for subsequent starts and stops. Thus, multipass membrane proteins are stitched into the lipid bilayer as they are being synthesized by a mechanism resembling a sewing machine.
That kind of cool. So the different signals,
Membrane proteins are
sequences tell them, kind of tells the translocation protein kind of when to stop translocating it. So then that is stuck into the membrane and then the ribosome continues its job. So, a video is so much better than me trying to explain this because it's kind of cool. Yeah. Kind of cool. So I would say 100% of membrane proteins start their lives like this. They're all made in the ER membrane because typically membrane proteins are not soluble. If you try to make them outside the membrane, they tend to stick together. Anybody have an issue with that in their research? So you have to, that's why they're hard to manufacture outside of the cell. So you have to make them in the membrane. Okay. Was that good, Jesse?
Everything told you.
Okay, so that kind of shows how you can get the different membranes in there. There's a dual pass membrane. That's pretty much it that you need to see. So full screen. Full screen. Full screen. There it is. Full screen. This one just shows that some proteins can actually help themselves translocate across the membrane. So they can have one domain that spans the membrane here, and then it helps to translocate the rest of the protein. It's how some pathogenic organisms get their proteins across the membranes. That one's kind of cool. Transport them around, called autotransporters. Okay, let's see, where are we on time? 8 minutes. Well, we could start with this a little bit here. Okay, so any questions on those? Basically, we're just looking at, so as translation is occurring from the ribosome, it can either be cytosolic or something like that, or it can be in conjunction with the ER. So if it's a membrane protein, it's going to be stitched. If it's some other protein that has to be modified, it's going to be translocated into the ER before it's being modified and moved around. Okay. So that's the point here. Okay, where do things go? Let's see. Let's look at vesicular traffic. So this is kind of a view of the endosome pathway. So here you have the ER, the rough ER. What we see is a lot of traffic going between the ER, the Golgi, and other membranes or other organelles. So a lot of proteins are going to be transferred between through these little vesicles. And we can see things going back and forth. So sometimes you can have proteins that are sent back to the previous areas. And of course, we have to figure out what these things look like, right? Because membranes kind of look the same if you're just looking at the lipids. So as it turns out, a lot of these other organelles are labeled with other proteins. So we can see things like clathrins or COP proteins. These are all called coat proteins. And they will basically identify the different types of membranes or endosomes that you're dealing with. So again, you have to regulate where things are going. A lot of this is going to be protein-protein interactions that are going to identify where the traffic is supposed to go and where these things are supposed to end up. So kind of cool. Let's see, how could they help? I kind of gave you the answer for that, didn't I? How can membrane proteins help to differentiate? How do they do this? So thinking about protein-protein interactions. Are they specific? I think that's where I was going with this question.
Is he texting his girlfriend again?
See, I shouldn't let you use your phone.
No, it's funny, this is marrying this.
That was the texting to go.
It isn't even bigger road.
And I know her, so she would come and yell at me. So, it's okay.
I mean, you do that, but I had to think the rest of it.
Okay, yeah, sorry. There's an inside joke, guys, sorry. Okay, right, they're specific, right? So you can imagine that there's going to be specific protein interactions between vesicles and other membranes, which are they're gonna help identify where these proteins are going, right? That's what I was trying to get you to think about with that question, okay? So yes, there's going to be specific things, and ooh, that's a good word, Sierra, facilitate, I like that, it's a good one. Good job. Okay, so let's look at some examples before we run out of time. So this is a kind of a classic example called a clathrin coated vesicle. So clathrins are these proteins on the outside, so they're going to coat the vesicle. They're going to interact with these other proteins. What are these? The adaptins and then the cargo receptors there. All those are going to function together to basically identify whatever the cargo is in that particular vesicle. So then once this vesicle buds off, and we use some cytoskeleton proteins to do this, then the clathrin coat is gone, and it's then the cargo proteins, which are then going to be responsible for moving these things around and identifying where they're supposed to go. Here we see another role for these phosphodienosyl proteins or lipids. So these things can be modified just like we saw in that PI3 pathway. It can be modified to affect which proteins bind to them. And we see different modifications in different locations. So this kind of shows you the colors for each of the different types of membranes. So here again, just think these things can be identified from the outside, okay? These vesicles are identified and the targets are identified and wherever they're supposed to go, those are identified as well, okay? So what we're gonna have is we have the cargo proteins there. Those are then going to interact with other proteins on the target membrane typically. And that's gonna lead to the fusion of that vesicle with that target, whatever it is. So if you think about, I don't know, a vesicle going to, could be the plasma membrane, right? That's a classic one. So the vesicle is going to bind to these proteins, these tethering proteins. And then these other proteins called snares, those are basically going to snare them and keep them or bring them close to the plasma membrane to help them then fuse together. I always got the T snares and V snares. I got that wrong on a test once when I was an undergrad. I was really mad at doing that. So the V snares, those are the ones on the vesicles, right? And T snares are on the target. Okay, so it's pretty straightforward. But the snares are kind of cool. There's actually, we'll talk about RABs here in a second, but here it is. The snares actually twist themselves together to bring the two membranes together. Once you get rid of all the water between there, then those two membranes will actually fuse together to form one membrane. So I don't know, it's kind of a cute couple thing, isn't it? Right? Those snares, they like each other a lot, so they bind and then twist around each other to bring those things together. Then they have to be, I think they're untwisted. If I remember correctly, they have to be untwisted after that. Okay, let's stop there. And then when we come back, I'll talk a little bit about these RAB proteins. The RAB proteins are going to be important in identifying where these vesicles are supposed to go. Okay. Have a great day. Okay. So we were talking about protein sorting. How do proteins get to where they go, right? So I told you that a lot of proteins have have signal sequences on them, which help them get delivered, and there's different ways to get places, right? You just have a membrane transport or a nuclear transport, or you can go through vesicles, and we were talking that vesicles actually have an address label of their own. Actually, all membranes do, not just vesicles. And so it's these protein interactions that are going to be really, really important for making sure the vesicle is going to fuse with whatever target membrane that it's supposed to go to. So again, right in here you have regulated, right? So it's a way to regulate how these vesicles are traveling, what they're going to fuse to, or even if they do fuse to things. Because if you look at neurons, aren't a lot of neurotransmitters just stored in vesicles until the action potential causes them to fuse, right? So that's a regulated step. You can actually store things in vesicles until they're needed. So what kind of proteins are involved? Well, I mentioned these tethering proteins here. So on the target membrane, you're going to have some sort of protein that's going to interact with the proteins on the vesicle, which are identifying what those vesicles are. Those identifying proteins are called RABs, and there's a whole bunch of different RABs associated with different vesicles or different organelles. You don't have to memorize it. But RABs, interestingly enough, RABs are kind of associated with these lipid rafts. Have you guys ever heard of that? A lipid raft? So normally we think that in a plasma membrane, there isn't much organization, right? It's a sea of lipids with everything kind of just floating around randomly in it, whether it's proteins or lipids or whatever. But what they have found are these lipid rafts, which tend to be kind of an organized little area in the lipid bilayer. And so they look like a raft, right? Or a boat where they kind of stick together. And the idea here is that you're grouping proteins which have some sort of associated function together. So by grouping them, maybe that helps the process go a little faster. I always think of those, what is it, like the electron transport chain or something? This would make sense for something like that, wouldn't it? I don't know if it's actually true, but to group certain proteins together to keep them associated. And so the RABs are part of this. And so they form a very important part of membrane fusion, where you keep these proteins together so that you can fuse these membranes very, very quickly, okay? So you don't have to memorize any of the RAB stuff, thankfully. But the idea is that the RABs are going to interact with these tethering proteins, and that's going to be that specific interaction you need to make sure the vesicle is going to go to the right place. Then, as those bring the vesicle in, then it's the snare proteins, the V and T snares. Those are the ones that are going to help actually fuse the membranes together. So the snares are kind of generic. The RAB proteins and those tethering proteins are going to be the specific interaction system.
So I see one snare on that little diagram. I can't remember if I have, I'm sure it's like a few snares.
Yeah.
Together. Does that matter? Does the number of snares matter?
Well, not on the exam, but it does. Actually, I have another picture. If you want, just give me a second. I think I have another picture of it. These things just give you an idea of how the RABs are activated. So again, they're highly regulated. They actually are associated with the GTP hydrolysis. So just like a G protein, they can be regulated in that instance. Here's the picture here. So there's two here shown. And the idea is they bring them so close together, you get rid of the water, and that's going to help speed up the fusion of those membranes. So to us, I'm not going to ask you any detailed mechanistic questions on this. What we're doing now is, right, we've looked at how proteins are made, and now we're kind of seeing how they're sorted and how, again, there's going to be other layers of regulation that we're looking at here. So now we're almost like we're stepping back a little bit, aren't we? Looking at more general processes than what we were looking at specifically. So another hand. Karen, is that you?
Yeah, I've heard it's like Q and R students, I mean, the same things.
Q and R?
And I've heard it, I said, Q and R, is that the same thing?
Oh, gee, that doesn't ring a bell. Is that something new that I've missed?
We talked about that last time too.
Q and R stuff? I'd have to go back and check. Sorry, those don't ring a bell to me. Sorry. I'd have to go check it out. Anyways, this kind of just shows you what they do. They're kind of cool. And of course, the snares have to be untwisted to be used again. So there are other proteins that will do that for them. Okay. So again, this was from a review paper, just kind of showing you the regulation of it, right? The more proteins we have here, the more we can regulate the process, because all we have to do is inhibit any of those processes, and then we can inhibit the formation or the fusion of these vesicles with its target. So I think it kind of goes along with any other process we've looked at in this class. Very highly regulated. Okay, so kind of cool. You know, I like pictures, so whenever I see cool pictures, I steal them from the interwebs, put them on there. Right. So, except what's my one mistake? I don't put the source. So I guess I'm plagiarizing everybody, right? So okay, cool. Let's see what else. Oh, look, snares can be used for bad things to happen as well. So here's an example of the HIV virion coming in, and it actually binds to a very specific protein receptor. You may or may not have known that already. So that's why it always targets a particular T cell, because it binds to that particular T cell's receptor. And then the virion actually has something that looks kind of like a snare. So that then actually helps it get into the cell all by itself. Isn't that kind of cool? So an HIV or a virus can do this as well. So what are the type of interactions that are necessary then? Kind of going back to what I said. Think about these. I'm giving you the answer. It's those protein interactions, right? That's what I'm looking at. You can be more. Again, if we don't have those specific interactions, then it won't happen. Right, specific interactions. You guys are so unique. Just say protein. You just take what I tell you, huh? Just take it. Whatever I tell you, you'll do it. Smart. I like that. Okay. Any questions on that? Kind of cool. So things you just have to keep in mind. So after translation, if these proteins are going to be sorted, there's a directory, right? The cell knows how to get things to where they're supposed to go through protein-protein interaction. Pretty straightforward. Okay, so what kind of things do we see happen? There are a lot of post-translational modifications as well. A lot of that happens in the Golgi apparatus. So the Golgi is this weird looking thing that has two different sides to it, a cis side and a trans side. And you can label things. You can actually see where those sides are. So again, even within this complex, you can differentiate between the different levels or areas of that particular organelle. And these are going to be spelled out again by the protein codes. So you can see the protein coding can actually dictate which side of the Golgi you're going to fuse with, and then which side of the Golgi you're going to come off of on the other side. So all that's going to be dictated as well. It's pretty complicated, right? So if you think about it, how would you describe the Golgi? Yeah, you guys remember that from my Gen Bio class, or is that from a different class? Huh?
Sixth grade.
Sixth grade. Wow, it's post office, right? That's how I always describe it. It's like a receiving area, and then, well, unlike the post office, they just fuss with your packages here, right? There's a lot of post-translational modifications that occur here. But I mean, how does that occur? That occurs with proteins as well. So interestingly enough, there's a lot of recycling going on as well. You might have proteins coming out of the ER that are actually recycled back to the ER for use. And the same thing in the Golgi. You might see these Golgi proteins recycled and reused. So there's a lot of transport going on. And of course, you have to know which direction you're going and where you're going and why you're doing that. So There's a lot of enzymes in here that are doing the modifications. I'll give you some examples of some of them. So here, especially in higher organisms, we see a lot of sugar additions. So, and that of course changes from species to species, but there's all sorts of things we can see. So phosphorylation, addition or removal of sugars like galactose or mannose or things like that. Of course, it's going to depend on the protein, right? So proteins are going to have their own unique modifications for whatever they do, but there's a lot of stuff that goes on there. There's also some proteolysis that can occur. So some proteins can be cut into pieces to be functional. So this is an example of a pigment protein that's actually cut into pieces, and then each piece kind of has their own separate jobs. If you look at what they do, where you don't have to know exactly what they do, but a lot of processing that can occur. Here's just another look at it. So again, especially in humans, you get a lot of sugars. What is that going to do to the protein? Does that change structure, function? Yeah, sugars are pretty polar, large groups. You can imagine that it's going to affect the folding or function of those proteins in some way. Especially for things like antibodies. As antibodies go through, there's all sorts of sugars that are added to those. So some proteins can be heavily modified. What does it say on there? 100 different monosaccharides used. So quite a variety. And these will differ, like I said, between species. I forget what it is for humans, but a lot of our proteins have a particular sugar motif. And then if you look at mice, they have a different sugar motif. So it can change. Okay, what else? So the Golgi is going to sort them depending on where they're going. So you can imagine that if things are going to be secreted, then they can be stored in these vesicles for a certain period of time and then secreted at some point. Some things are secreted all the time. So you can see those just going and fusing with the membranes. I think that's pretty much it. And then some, of course, can be shuttled somewhere else. So these are not the only places it's going to go, obviously. What kind of things do we see? Do we see any unique vesicles that are made for particular purposes? Very leading question. Yeah, so think about digestion or lysosomes, something like that. So this is also a way that we can make specific types of vesicles for a particular job. And the lysosome's job is to fuse with something and digest it, right? So you see a lot of digestion enzymes in here. It's probably a good reason to keep them sequestered inside a vesicle and not just floating around a cell as well, is to put them in there. But they're pretty complicated. Here's a cool picture I got off the internet. I don't know. This gives you an idea of how complicated it can be in terms of the membrane proteins, as well as the cargo that's inside. And I don't remember exactly what that is. Maybe it's a lysosome or something, but pretty cool looking. So what are these things? What can they do? Well, if we use something like endocytosis, that's the process where you take a big gulp of something from the outside and bring it in. We can see those fusing with lysosomes, and then the lysosomes typically are going to chew up whatever's inside. In this case, they're chewing up cholesterol to use for other processes. And then you can either recycle the receptors and make more lysosomes as well. So this just kind of gives you an idea of all the traffic, a lot of different traffic on there. Have you guys ever seen that video of inside the cell, and they show this little vesicle being pulled along the cytoskeleton? That's kind of what you think of. Sorry,
By the motor protein.
yeah, by the motor proteins, yeah. And we'll talk a little bit about motor proteins, but not much. But it's kind of cool. I just always think these things are on highways, basically. They're being moved to different areas. You can also think of the neuron, right? How do we move things from the neural body down to the synapse along that long axon? Yes, cytoskeleton, right? It's like a highway. So what you say. Yeah, it's like a highway where you can pull these things from one side to the other. So it's pretty cool, pretty easy. Well, not easy, but a pretty convenient or efficient way to transport things from one part of the cell to the other. And then this just gives some ideas of what happens to different receptors. So if you're binding things in these transport vesicles, you're gonna also recycle the receptors or degrade them or move them other places. So this kind of gives you an idea where these receptor proteins are gonna go. And then another view of these lysosomes binding and chewing things up. I think that's pretty much it. Oh, and then there's the picture of moving things around. It would be really difficult to just have the vesicles from this side diffuse randomly over the synapse, wouldn't it? It would probably be a low kind of percentage process. So having that particular movement from one side to the other is going to make it a lot more efficient. Okay, I think that's pretty much it.